I'm actually presenting this work on behalf of a PhD student in our lab. Yara Ujaimi, she couldn't be available today, so this is part of her PhD work. I'm a postdoc in the lab working with her under the supervision of Dr. Ellen Blasco. So there's three axes to this project. The first is the use of single chain variable fragments, which for those of you who are not familiar, they're basically variable regions of an antibody's heavy and light chain that are put together, that are bound together through a peptide linker. And these are basically biotherapeutic agents that are commonly used. And in the first part of the project, we wanted to take these scFvs and screen them against a TDP43 proteinopathies, clone them into vectors, and then look at their effect in vitro. In the second aspect of the project, we wanted to characterize a new mouse model for TDP43, and look at different aspects in characterizing this model, including different Western blot analyses, spinal cord MRI, metabolomic analyses, and in progress is the analysis of the gut microbiota. And in the third part of the project, we wanted to basically integrate these two approaches and use AAV capsids that are enclosed with the scFvs, and then incorporating this biotherapeutic agent into the into the mice that we characterized. So just to start with some data that we've collected from the first branch. Using phage display technology, we wanted to screen the scFvs for TDP43, through which we found two scFvs, which we named B1 and D7. Through an indirect ELISA assay, we were able to confirm the binding of these two scFvs to a wild-type human TDP43, which, and in the control group, we found no evidence of binding. And then given that there's no 3D model for TDP43, we used a predictive in silico technique to look at how these scFvs were binding to TDP43. And what was found was that B1 was bound mostly predominantly to the RRM1 region and glycine-rich regions, whereas the D7 scFv bound to the N-terminal and the RRM1 and 2 regions. And then we wanted to take these scFvs and co-transfect them into hex cells that already overexpress TDP43 as an in vitro model for ALS. And in the left panel, we're looking at the localization of scFvs with TDP43 in regular hex cells that did not overexpress TDP43, and we find that both B1 and D7 localized with cytoplasmic TDP43. And then on the right panel, we are looking at the hex cells that were overexpressed with TDP43, and we find that the scFvs also co-localized with TDP43 that was mislocalized and aggregated in the cytoplasm. MTT assays were also done to look at the toxicity of both scFvs and the overexpression of TDP43 in hex cells. However, no evidence of toxicity was found. Now, given that TDP43 in ALS, we do know that there is fragmentation into 35 and 25 kilodalton fragments, we wanted to see how these scFvs affected the fragmentation of TDP43. And data that you're looking at here is from the soluble protein fractions that were collected from the hex cells. And what we found was that there was no effect of B1 or D7 on the soluble or full-length TDP43, suggesting that the functional TDP43 should not be affected. However, once the D7 scFv was co-transfected with the HEC cells, we found that it was able to actually decrease the C-terminal fragment at 35 kilodalton through a mechanism by proteasomal degradation. Since it's not very clear, but once the MG132, which is the proteasomal degradation inhibitor, was added to the experiment, we found that this inhibition was removed. And given that B1 didn't really show much change in the effect on TDP35, it was later on removed from future experiments. So here on out, we're looking at basically D7 data, and we wanted to see how D7 affected metabolomic changes. And this is especially important because we do know in ALS, it's been documented highly at this point, that there are metabolomic alterations that occur in ALS patients. And what was found was that in this in vitro model, D7 was able to bring back to baseline different lipids, including phosphatidylcholine, palmitoyl carnitine, and hydroxyphenylglycine. And this is important, again, because these specific lipids were highly documented in many studies to become altered following in patients with ALS. So that's a very brief summary of some of the in vitro data that was collected and how scFvs altered the TDP43 profile. And then we wanted to move on to a novel mouse model for TDP43. And this is a model that we were able to obtain from GEM Pharmatech Labs in China. And this part of the project was done in collaboration with our industrial partners at Kiobs in Orléans. And basically, this is a novel model for TDP43. Nothing's been published about it, and we had the opportunity to work with it. So we, first of all, wanted to look at the phenotypic characterization of this mouse. What happens at the level of motor control? What happens at the level of body weight? And how is this altered in this TDP43 model? And this is data that was collected over a 10-month period. So the first thing that we're looking at is that there is actually an evidence for a decrease in body weight, which is evident in all TDP43 mouse models. And then, of course, we want what's more important is the motor control. So here we've got data from the hind limb grip strength. We've got data from the rotarod and footprint test. We've also got a combination of other assays that were done, but for time's sake, I'm keeping it short. But what we see is that there's this very nice decrease in motor activity for the TDP43 mice. But the rotarod test shows a very nice decrease across a 10-month period, which continues to decline. Similarly for the footprint test. So from there, we wanted to see how TDP43 was actually expressed in these mice. We did a Western blot analysis, and this is still underway. We're still waiting for some data as far as different tissues are concerned. This is data collected from the brain. And we find that there is an increase in TDP43, but it's not significant. However, that's not to say that there's no actual effect of the protein on the phenotypic data that we're seeing. It could just be there's changes in the post-translational modifications of the protein or increased C-terminal fragments that are happening, but we're still looking into that for more detail. We also did some metabolomic experiments and lipidomic experiments, some of which is still being analyzed, but essentially when it comes to the metabolomic analysis, over the 10-month period, we do see this nice distinct separation of metabolites for the transgenic and wild-type mice, which was maintained over the 10-month period. Moving on to the third aspect of the project, which is still also underway. So this part of the project was actually funded through Sanofi for the IdeaTech Award project, which was given to Dr. Hélène Blasco. And essentially here we're looking, so this is a pilot study that was done. We're actually starting the actual project in injections this October. But what we did here is that we wanted to take these scFvs, we know that they work in vitro, and we wanted to see how they would affect changes in the in vivo model for the GPT mice that we are aiming to characterize. And so the first issue that we had to deal with was how is the scFv and where is the scFv expressed if we inject it into these mice? And we had to go through an entire array of experiments before essentially we found success with IV injections of the scFvs that were enclosed with the AAV CAP-B10 plasmids. And we injected them for over a 4-week and an 8-week period. Both timelines gave us very similar results with respect to the expression of the His tag, which is what's bound to the scFv, so that we know that they're expressed. And what's nice is that we found expression of the His tag for both D7 and B1 to be fully localized to the central nervous system. Whereas we don't find evidence in the kidney or the liver or the muscle, suggesting that there's no risk of hepatotoxicity or inflammatory response. We also found, with respect to mRNA expression, that very similarly, we only found expression to be localized in the central nervous system. So just to sum up, essentially what we're looking at here is to look at a therapeutic approach to target TDP-43 proteinopathies using scFvs and evaluating their effect, their toxicity, at a both peripheral and central level. And to do that, we're using a novel TDP-43 model, which we're aiming to characterize, and looking to see if, at the end of the day, there's any advantages of it over other TDP-43 models that are currently being used in the research. And then the use of omics to study the effect of drugs and biotherapeutics. And that is the team that I work with. And on behalf of Yara, thank you. So if there are any questions.
Thank you. Questions?
Hi. Just a technical question for the new mouse model that you use. So I didn't understand the wild type. Is it a knock-in human wild type, or is it a double transgenic?
I do believe it's a knock-in human wild type. We don't have all of the data that we would like from Gem Pharmatech Labs, so we are still looking into more detail about the strain and how it was derived. We don't have all of that information yet, but we are in contact with them to obtain it. But I do believe it was human wild type.
Okay, because when you look at the level of expression of TDP, that would make sense now if it's a knock-in, that you would not have the overexpression if you compare just the knock-in to the... I don't know, I was confused.
Yeah, I'm not sure as far as to the expression of TDP-43. We thought it was a little weird also at first. We do require some more information from Gem Pharmatech before we can answer that question. But it's not to say that we could, because we do see a very strong effect on motor control. And we are aiming to characterize the model further. But we're going to wait and see what happens. Yeah.
Thank you.
So I'm here.
Yes.
I would like to follow up on that. So the mouse model, is it expressing human TDP?
I do believe so.
Okay, so it's not a knock-in, or it's a knock-in of humanization. Okay, no, okay.
I am not sure.
Because it's important for your
Yes.
antibody, because it's a human antibody.
Yeah, yeah.
And my real question was another one. So did you test whether when treating your cellular model with your antibody, your scFv antibody, did you test whether you modify TDP-43 function, for instance, cryptic splicing?
For example?
Cryptic splicing.
I don't believe that was done. this was a little bit before I joined the lab. I don't believe that that was done, though, no. Yeah.
Thank you.
I also had a question
Yes.
here, because a lot of the data with the antibodies, the intrabodies, were on TDP43, I imagine, wild type.
Yeah.
Did you ever test the mutant, any of the mutants? Because they obviously have more of a propensity to aggregate and to go in the insoluble fraction.
I'm not sure. That would be Yada's part of the project. I'm not able to answer that. I'm
Thanks.
sorry. Yeah.
Thanks, Samira.
Thank you.
Okay. Oui.
Thank you very much.
Good luck.